now we're going to proceed with the questions. And after this, we break for some coffee. It's going to be here to the left side. So, if there's any question, we have the microphones. So feel free. You have a question?
Oh, so thank you so much. That was absolutely fascinating. I'm a parent of a GRIN 2B probable loss of function, and just with respect to some of the notes that you were presenting, Dr. Wyllie, when you looked at, say, things like pain responses, etc., did you look at hormone levels for those kinds of things, especially with hippocampus long-term memories? Because I've noticed myself, like, big experiences are remembered.
Okay, so we haven't, and I haven't shown all the data there. That the major effect does appear to be on the tactile rather than on the pain response in the GRIN 2B model. So it's the tactile, and that's also quite common, as I say, and alluded to in other models, in the SYNGAP1 model as well, which is, again, it's kind of an NMDA related one because SYNGAP is a protein that NMDA receptors signal through. So it's perhaps not surprising that we see these types of responses there. Did you ask about hormone? Did you say about hormone?
Yeah, I was just wondering if you had recorded or looked at those at all.
We haven't really recorded this, but I think it's a fantastic question and really highlights where we need to go in terms of a field to have a much better integrative approach to the study of this. And you know, I'm not forcing Alfredo to talk about this or anything like that, but the types of experiments that Alfredo's looking at in terms of overall and studying at a much higher level, I think, asked that. And we've got a colleague, Sally Till, who's working also on various models, who's looking at what happens to heart rate, what happens to, in terms of what we're measuring as a brain, what we are measuring as a behavioral, brain activity. But this integration across is absolutely key. And there's a huge amount of literature in terms of signaling from the periphery to the brain and the brain to the periphery, ideas about, gastrointestinal problems, so the so-called gut-brain axis. You all know in terms of how diet can affect the behavior. So we need to get better. And what we are looking at here largely is what we might call a reductionist point of view. We're looking at individual cells, single cells, or a very, very simple circuit. And hopefully, as I explained, that the brain is way more complex than just cutting out a little bit of brain and stimulating and looking at a few of these synapses. So we need to get better at that integrated approach.
Okay, thank you. That's fascinating.
Let's say one more thing, and I think this is exactly what we want with the associations, is that you have access to that information. And this is where more collaboration with the associations is best, because we can put out questionnaires and have family exactly respond to those blind spots that you say, how do I translate this from a rat model to a human being?
Yeah. I was a bit cautious about this because I wasn't entirely sure how best to approach this, but I'm very happy to liaise with you and ask the questions, and if you want to send it out to the community as a I don't say a formal questionnaire. I'm not asking for private medical. It's more anecdotal, just to help inform our experiments. But I would be happy to chat to you about
that. We collaborate with all the associations, so the idea is to get it out to all the community. But thank you. Sorry. Next.
Hi, I have a question here. Dali. Hi. About the compensation mechanisms in the both Rett GRIN 2B and GRIN 2A haploinsufficiency, is there work on some sort of analysis like RNA sequencing that might give us some light about the molecular events that contribute to this compensation?
So that's a great question, and you're right to say to highlight the potential for compensation, because what we are looking at in terms of our models, which was a little bit different from actually some of the other models where we're introducing a protein with a mutation in it at a stage where there has been development ongoing. We're looking, these rats can, have the mutation from birth. So there is a lot of compensation that potentially can go on. We have not looked at in terms of where the absolute the compensation can be, but in terms of GLUN2B, there is GLUN2B is my idea about GLUN2B is actually the most abundant NMDA receptor subunit in the forebrain, in the cortex. And perhaps part of the lack of effect is that there is we like to think of it as a bit of a reservoir of 2B there that can compensate. But equally, and again, I'm not trying to give any spoiler, Alfredo is going to show you some very different effects in terms of GRIN2B, where there are very dramatic effects at a circuit level. Okay, so I think that what we're looking at, we're sort of at the step one, right, of doing this. And the way that I normally explain it, so I flew in from Edinburgh yesterday, so I had to get to the airport. So that's my first step, was getting to the airport. And then I had to get a flight, but I could have taken a flight, rather than taking, it was Ryanair, from Edinburgh direct to Barcelona. I could have flown BA and gone BA to London and London to Barcelona. And that difference there in terms of how the brain communicates between the regions may well be, the differences. So we're only looking at individual steps and not necessarily at all these circuits and how the circuits interact with one another.
Thank you all. Great talks. This one is actually a question for Olivier. So when you remove the carboxy terminal domain, most of it, as you present in that 1004 mutation, nonsense mutation, do you see any, so you completely lose the ER retention motifs of GLUN2B. So have you ever tried to do experiments whereas you do not provide other subunits or in other systems to see whether that can actually does escape to the membrane, anywhere in the membrane?
Not really. So what we're removing, it's most of the part of the C-terminal domain that we remove is related to the scaffolding protein and the signaling protein. So for the formation of the NMDA receptor, like addressing at the surface, I think this is completely normal because we see the receptor, but not really, we don't know where the receptor is going when it's escaping the synapse, for example. So it's around, and after probably is trapped, and there are some processes on those cytos. So we don't know yet what is the consequence of this hypermobility for the brain function, at least for the receptor function. I'm not sure that I answered.
Yes, I had also a similar question as you did for Olivier. So it was regarding the number of GLUN2A or GLUN2B containing because of these changes of the mobility. And I have also a couple of questions about the rat models. So you were characterizing this electrophysiological signature of the model, and you were considering the temporal summation. But perhaps this discordance with what you observe could be explained by spatial summation fact or by changes on the synaptic numbers. If there is any kind of homeostatic plasticity mechanism that could be compensating these changes, and perhaps this is a mechanism that could be explaining. And second, I have also, we have been working in the lab a few years ago with the GRIN2A mouse model, the knockout one, and we found some similarities with some of the models that you are commenting. So first, we also observed somehow a band of in the Western blot, a faint GRIN2A expression even in the knockout. So that was a bit impressive in our hands with the mouse model. And second, when we received the mice, they also exhibited a very crazy behavior. They were super excited. They were jumping out of the
2A or 2B?
The 2A. The 2A. I mean, yes, but the point is that when we crossed with external mice or breeders, then after 3-4 generations, we just removed this genetic drift, perhaps, this effect. So that was something that at the beginning we were also thinking about something that was very related to the limbic system or emotional responses, super stressed mice. But then it came to light that it was not that point. So what is?
To take that last point first, our rats are actually are outbred models. So we try and ensure that we've got a lot of, we're basically losing what we're studying is the loss of 2B and not a sort of genetic drift in the sense of the inbreeding from that. I don't know if you were alluding to the fact that was the genetic background of the mouse rather than the loss of 2A.
Yeah, perhaps my genetic background is not that good. No, but it was related with the when we received the founders. We just received four founders.
Yeah.
So then we were crossing, and at the beginning they were just like very strange behavior.
I see. Okay.
Yeah.
So we again, I'm not going to steal Alfredo's thunder, but he's going to show a lot more data in terms of I presume seizure-like activity in GLUN2B. It is less so in 2A. We really don't see much evidence for seizure-like behavior in the 2A rat model. Getting back to the summation point, I appreciate this is getting technical. I'm happy to follow up with you later on. I understand the idea about, that it could be, homeostatic plasticity, but what doesn't get round is the fact that, why is the actual profile of the responses when we've completely lost 2A, we would expect them to change, and they're not changing. So that is one that doesn't quite fit with the homeostatic plasticity. But I take your point about that could be a potential, but we've done a lot of control experiments to try and unveil and unmask the NMDA component. And if you actually read the literature, there's some very old literature looking at NMDA synaptic potentials in the spinal cord, but not a huge amount in terms of the brain hippocampus. So we really try to go back and address this, because it is an observation at the moment that we're struggling a little bit to understand.
Thank you.
May I ask a question related to the rat knockout mice, the GLUN2B heterozygote? So you don't see any modification of the recent memory formation, but did you try remote memory? Because there are some
Sorry, which memory?
Remote memory, the long-term memory.
So the only memory, as far as we've gone, I think we've done. I showed the 24-hour memory, and that was not impaired. I'm trying to remember if Lysandra's, gone beyond that, but that was certainly the observation is that the 24-hour memory was not different.
Yeah, but just to comparing the data that the Paul Franklin find on the remote memory, the long-term memory, found that, for example, for the heterozygote knockout mice for CAM2 which interact with the GLUN2B receptor, you don't see any impact on recent memory, but you have an impact on remote memory based on the fact that the modification, the plasticity at the cortical level, it's affected, but not the one at the hippocampal level.
Yeah, we haven't looked at that.
Very nice talk. I have one question for Andy and one question for David. For Andy, the two gain-of-function variants you transfected actually pretty mild.
Yeah.
It's a one for the compared wild type, the chart transfer, the other one is 1.94. There are 2 or 3 dozen of the strong gain-of-function one, 20, 54 gene. Do you think if you transfer any of that strong gain-of-function one will dominate the output?
That's why we asked you for those constructs.
Okay.
Yeah. So when we started this was around 2000. When we started actually deciding which variants to look at, it was 2017 or so. So this was that was around the time when a lot of the variants that were kind of identified were the sort of agonist binding domain variants, including those. And so we picked the strongest ones that we could see that at the time. And obviously since then there have been some stronger gain of function. I mean, they do, at least in sort of the parameters that we've looked at, they look kind of gain of function. But you're right, it's not super striking. So what we could do is we could see to what extent what we're seeing with those, we would see with stronger gain of function variants, which are typically more kind of ion channel variants. Yeah.
Thank you. For doing very nice work. Do you think the high level of anxiety confound your water maze memory test? Since we have knock-in mouse-like hosting loss function GRIN 2B variants, which is highly access. And that we also did water maze, we did field conditioning, but we could not tell any difference. But when we do some others, like less stressful test, it can tell the difference. My question is, do you think the high level of anxiety confound the learning memory test?
I mean, it could. The fact that they are not different from wild type in terms of the test, you would need to argue that the level of anxiety is then the compensation for the memory task, I think. So I don't know. We haven't looked at, and I'm trying to think what we've done with the fear conditioning. Alfredo, remind me, we've done fear conditioning in GRIN 2B, and it's. So I think there is a difference in auditory. I can't remember the way it goes with the auditory fear conditioning, and certainly in terms of we're trying to sort of look at ethologically, relevant behaviors for the rat. So we actually have a prey capture test in terms of GRIN 2B. So we basically get the rat to catch a cricket, right, an insect, and we put it in and try and get the rat to catch a cricket. And actually, the 2B hets are actually very much, well, a lot of them are just not interested in actually trying to capture it. So, but if the ones that do capture, they will sort of capture, I think, normally, but a lot of them just show disinterest in trying to capture that. So we can see behavioral, phenotypes in the model, but not in the more standard ones. And I guess we looked at the previous ones because of the sort of historical view of 2B and 2A roles in plasticity in terms of potentiation and depression. That was our sort of starting point on that. But there's a lot more that we need to do in refining these and certainly addressing the potential anxiety issue in that. But I think if we separated out the males and females in the water maze, we didn't see any sex difference there.
Okay. By the way, we also did the pain threshold test. Or knocking mice. We could not tell the difference between the wild type mutant, which is consistent with your data.
Yeah, that's the mouse.
Mice.
Yeah.
Thanks.
Sorry, we have time for 2 more questions. So I know there's already a mic over there.
Yeah. Hi. Now, my question is on the connection between SYNGAP and NMDA receptors, because as far as I know, most of the knowledge has been developed on the connection between SYNGAP and AMPA receptors is another expression. So could you comment a little bit more on what happens in SYNGAP with NMDA? Thank you.
So SYNGAP is a protein that's involved in so the C terminus that Olivier was talking about is a massive signaling complex, and SYNGAP is one of these proteins that is found in this signaling complex. And the signaling, you're absolutely right, the signaling from the NMDA receptor through SYNGAP actually manifests as a change in AMPA receptor expression. So it is AMPA receptors in certain models that are affected, but the signaling is coming from NMDA through SYNGAP and then affecting AMPA. So that's why you're absolutely right. What is often measured is the AMPA receptor function and how that changes in these models, but the signaling is not coming from the AMPA receptor directly to SYNGAP, if that helps.
If I can add something, that in the lab we are also studying the mutation that touch the protein that interact with the C-terminal of the receptor, of the NMDA receptor, and we can try to see what is the impact of mutation of intracellular interactor on the receptor mobility and trafficking. So it's not only about the C-terminal domain of the NMDA receptor or AMPA receptor. It can be also have the same consequence if you touch intracellular interactor specific to each cell unit.
Yeah. Oh, yeah. So thanks for 3 great talks. I had a question for Andy about the T-kit approach. So as I understand it, you're starting with a wild type background, and then you're just trying to introduce the variant at a slightly later stage. Is that correct? Or a slightly earlier stage than you'd previously been managing in the culture?
Yeah, that's right.
Have you considered trying it the other way around, where because obviously that would give you normal development, and then you're looking at kind of variant receptors on top of that normal development. Would it be possible to take a knock-in line and then rescue wild type receptors onto that line? So you kind of swap the experiment around. You're looking more at the changes in development rather than the changes in the mature synapse or more mature circuitry.
I mean, that would be interesting to look at kind of how the sort of non-cell autonomous kind of effects of the variant. And I think some of the reason why the variants that we've got on our to-do list now are variants which we know people have got mouse models of, so that we can get some data using this approach in a wild type background, looking at sort of modeling the variant on a sort of individual neuron basis. But then we can think about experiments like you're saying, kind of collaboration, and even look at kind of implications for behavior. We've got people we can already sort of talk to and sort of show them, oh, look, this suggests that there might be some effect on, I don't know, Bradil or something during development in your mouse model. Is this something we could look at together? So, yeah. I did have a question actually following up from a question.
Yes.
About the summation experiments, and I just wondered to what extent calcium activated potassium channels in this sort of slower decaying, and so more calcium coming in through the NMDA receptors in the GRIN2A deletion context, could that be sort of dampening down the integration, the depolarization in those cells?
It could be. In terms of the actual profiles of, much of what Daniel's done, the data I didn't go into much. We don't see effects. There is obviously that is the sort of compensation type argument, but I still struggle with the idea. And this is maybe a very granular level of trying to understand function as to why, when you completely lose the fast NMDA receptor subunit, you do not see any effect on the EPSP. And that I don't think can just be explained by compensation.
Thank you. So let's go to the break room. We're gonna have some coffees and patisserie, and you can please serve yourself. We need to be back here at 11:30. If we could try to be as on time as possible, because we're having people join online, so we don't want them to wait. But thank you again very much to our speakers in this blog. Thank you.